In this lesson we're going to be looking at osmosis and active transport. Now the AQA specification has this to say about that. Water may move. Diffusion is the net movement down a concentration gradient. This process allows small molecules such as oxygen and carbon dioxide to cross the plasma membrane. Most polar molecules, such as sugars and proteins, can't freely cross the lipid membrane. Although water molecules themselves are polar, they're small enough to pass through the cell membrane freely. This special case of diffusion that involves the movement of water molecules across a semi-permeable membrane is called osmosis. If a molecule, such as glucose, is added to one side of a membrane, it will not be able to cross the membrane because it's both large and polar. Because of its polar nature, it will interact with other polar molecules, such as the water. This interaction reduces the number of freely moving water molecules on the right-hand side, and with fewer freely moving water molecules on the right-hand side, there's now a net movement of water molecules down their concentration gradient to the side with the glucose molecules. And because more water molecules are moving into this area than are leaving, the water level on the right hand side will rise. So, the definition to learn. Osmosis is the passive overall movement of water from a dilute solution to a more concentrated solution through a partially permeable membrane. Now remember, a more dilute solution has lots of the solvent, that's water, and less of the solute. Plant roots take up water by osmosis. In the soil, the water contains only a small amount of dissolved minerals. In the plant roots, there are lots of dissolved minerals and sugars in the cytoplasm. Water moves from the soil and into the more concentrated solution in the roots, and this is through a partially permeable membrane. Once in the plant, the water is then carried up through the xylem vessels, through the stem and out of the leaves. This helps to maintain the concentration gradient, and it allows the water to move into the plant continuously. Plant cells rely on osmosis to keep them packed full of water for support, which is why they'll wilt without water. A well-watered plant has cells that are so packed with water that they're swollen. They're described as being turgid. As water enters the plant cells by osmosis, the cytoplasm is forced against the rigid cell wall. Cellular cell walls can bend, but they don't stretch easily, so the cell won't burst. But the pressure inside will rise, and this is known as the turgor pressure, and the cells packed with water are referred to as being turgid. If the plant doesn't have enough water, the cytoplasm shrinks and the cells lose their turgor pressure. At this point, the cell is described as being flaccid, and the plant will wilt. Now these onion cells have been placed in a very concentrated solution of sugar. Water is leaving the cells by osmosis, and eventually the cytoplasm is going to shrink so much that it will pull away from the rigid cell wall. At this stage, the cells are known as being plasmolyzed. Now you can use this effect to investigate what's going on inside the cell. You might be shown images of plant cells held in different conditions. If you plot the percentage of plasmolyzed cells in each, when it reaches 50%, then the solution outside the cell has the same concentration as the average inside the cells. You could also do the same thing using weight. A potato placed in water gains weight because of the water. Potatoes placed in strong glucose solution lose weight because they've lost water. Now if you draw a graph of that effect, plotting the concentration of the solution against the percentage weight change of the potato, you can find a concentration somewhere on the graph where the potato just stays the same. Doesn't lose, doesn't gain water, stays exactly the same. Now at that point, the concentration of solutes inside and outside the potato cell is going to be the same. There are some very useful terms for describing these situations. You won't be asked for them directly in the exam, but your explanations will be far more impressive and easier to write if you learn these quite complicated definitions. The strength of the solution surrounding the cells refers to the amount of dissolved solute that they contain. And with lots of solute, they are hypertonic, and with less solute, they are hypotonic. Now, they're very similar, so it can be quite difficult to remember these terms. A way that I use is to think about hypodermic syringes. They go into the skin. So hypo means under. So hypotonic means less solute. Being very excited is sometimes known as being hyper. So hyper means more. Hypertonic means more solute. Just remember, it's all about the solute, not the water molecules, and then you can work out the rest. So cells in a hypotonic solution have less solute around them and more water around them. So there's a higher concentration of water molecules outside the cell. Water molecules move by osmosis into the cell. Now, animal cells, like the red blood cells shown, don't have cell walls. If they take in too much water, they can burst. That's called lysis. Animal cells need to keep the levels of dissolved molecules such as minerals and sugars quite constant. If they don't, then the cells can become damaged when they change shape too much. Moving on to active transport. Living organisms can't always rely on the passive drifting of molecules as happens in osmosis and diffusion. Some substances need to be taken from an area of low concentration to an area of higher concentration against the concentration gradient. Now that uses energy, so it's referred to as active transport. Now here's another definition to learn. Active transport is the overall movement of particles across the cell membrane from an area of low concentration to an area of higher concentration, using the energy released from respiration. An example would be the mineral absorption by root hair cells, or, as we're going to go on to look at, the absorption of sugar from human intestine into the blood. Now, there isn't just one method of active transport, there are a few, but this animation should give you a good idea of how it works. And the three main things to notice are: there's a protein spanning the membrane which is doing the transporting. The protein has got a complementary shape to the particle that it's moving. The process uses energy, and it's used so that transport can go against the concentration gradient. The energy is needed to change the shape of the protein, to make it move the solute through the membrane. Now, the fact that proteins with a specific shape are involved is important. It means that anything which affects the shape of proteins, such as heat or pH, can affect how well the transport can take place. And since it needs energy, anything that affects the process of respiration will also affect how well the transport can take place. So the cells involved in active transport will therefore have more mitochondria than normal, and a supply of glucose and oxygen will be needed for them to function properly because of the need of respiration and the energy it produces. Or releases, sorry, never say produces, it releases it. Since the cell needs a specific protein for each specific particle being actively transported, it means that the cell will only be able to actively transport particular molecules, and the speed that it can absorb them depends on how many of each type of protein carrier it has in its membrane. Now, the two examples of active transport mentioned in the specification that we need to know about are, firstly, plants absorbing mineral ions from the soil. The plant cells have more ions than the soil, but the plant still needs to take the ions from the soil if it's going to grow. An interesting consequence of this you may need to explain is that most plants don't grow well in waterlogged soil, or soil that has been compacted by trampling. When air spaces in the soil are made smaller or filled with water, then there's less oxygen available to the root hair cells, and the oxygen is needed by the root hair cells for respiration, which is used to power the active transport used to absorb the minerals from the soil. Second example that we need to know about. Epithelial cells lining the gut in humans need to absorb glucose from digestion into the blood, but they must also prevent the reverse flow of glucose from the blood into the gut. Now, using active transport means that glucose always flows in just one way, into the bloodstream, no matter what the gut concentration of glucose is. If you think about it, if we relied on diffusion, then when there was no food in the gut, glucose would leave the blood and flow into the gut, and that would be wasteful and probably even lethal. So it's worth the additional energy cost of active transport to make sure that glucose transport is just a one-way process. Now, there are lots of other examples that you might come across though. Fish living in seawater take in excess mineral ions and have special cells in their gills actively pumping the ions back into the water. And in humans and our kidneys, when the small molecules are filtered out of the blood in the kidney, we use active transport to retrieve the useful molecules that we need to stay in the blood. Don't forget to download the resources for this video from the link in the description, especially the notes template. LearnFix has a lot more to offer than just the videos and the exam questions. The most popular aspect is the set of 12 online learning games we have for each topic. These help you practice your recall of the details. Now, when it comes to revision, LearnFix is there to help. After each section of the syllabus, there's a diagnostic quiz to help focus your revision efforts. You can then use a progress report to look back at the details for that particular topic, what went well and what needs more work. We're adding new features every month, so visit us at learnfix.com to find out more.